Hello friends, today we will learn about growth promoting bacteria in plants. So, let's begin. Certain beneficial microorganisms present in the soil are known to influence the plant growth, development, and yield. These bacteria and fungi may provide growth promoting products to plants or inhibit the growth of soil pathogenic microorganisms which hinder the plant growth. The former is a direct effect while the latter is the indirect effect of growth promoting bacteria in plants. The growth promoting activity of microorganisms and the bacteriological approaches are: first is biological nitrogen fixation, second is biocontrol of phytopathogens, third is biofertilizers. These are the three growth promoting microorganisms. Okay, so let's start with biological nitrogen fixation. Biological nitrogen fixation. Nitrogen is an essential element of many biomolecules, the most important being nucleic acids and amino acids. Although nitrogen is the most abundant gas, it's about 80% in the atmosphere, neither animals nor plants can use this nitrogen to synthesize biological compounds. However, there are certain microorganisms on which the living plants are dependent to bring nitrogen into their biological systems. The phenomenon of fixation of atmospheric nitrogen by microorganisms is known as diazotrophy, and these organisms are collectively referred to as diazotrophs. Diazotrophs are biological nitrogen fixers and are prokaryotic in nature. Now let's know about nitrogen cycle. Nitrogen enters the soil with the deposits of dead animals and plants and urea of urine. These waste materials, that is proteins and urea, are decomposed by soil bacteria into ammonia and other products. The ammonia is converted to nitrite, that is NO2, and then nitrate, NO3, by certain bacteria belonging to the genera Nitrosomonas and Nitrobacter. The nitrate is degraded by various microorganisms to release nitrogen that enters atmosphere. This atmospheric nitrogen is taken up by the nitrogen fixing bacteria and used for the synthesis of biomolecules, that means amino acids. As the animals consume the leguminous plants as food, the nitrogen cycle is complete. Now let's know about nitrogen fixing bacteria. It is estimated that about 50% of the nitrogen needed by the plant comes from the nitrogen fixing bacteria. These are two types. There are two types of nitrogen fixing microorganisms. First is asymbiotic and another is symbiotic. Now let's know about asymbiotic nitrogen fixing. The gas fixing microorganisms. The gaseous nitrogen of the atmosphere is directly and independently utilized to produce nitrogen rich compounds. When these non symbiotic organisms die, they enrich the soil with nitrogenous compounds. Several species of bacteria and fungi can do this job. Clostridium pasturanium, Clostridium pasturunum, Azotobacter crocum. The mechanism of nitrogen fixation by asymbiotic bacteria is not clearly understood. It is believed that the nitrogen is first converted to hydroxylamine or ammonium nitrate and then incorporated into biomolecules. Now let's know about symbiotic nitrogen fixing microorganisms. These microorganisms live together with the plants in a mutually beneficial relationship phenomenon referred to as symbiosis. The most important microorganisms involved in symbiosis belong to two related genera, namely Rhizobium and Bradyrhizobium. These symbiotic bacteria, also referred to as nodule bacteria, are gram-negative, flagellated, and rod-shaped. The host plants harboring these bacteria are known as legumes. Soybean, peas, beans, alfalfa, peanuts, and clover. Each one of the species of Rhizobium and Bradyrhizobium are specific for a limited number of plants which survive as the natural host. It is now clearly known that these bacteria do not interact with plants other than the natural host. The relationship between the symbiotic bacteria and the legumes is well recognized. On the roots of legumes, there are a number of nodules, that is swelling, in which rhizobium species thrive. These bacteria trap atmospheric nitrogen and synthesize nitrogen-rich compounds, that is amino acids, proteins, etc., used by the legumes. At the same time, the legumes supply important nitrogen compounds for the metabolism of rhizobium. The growth of legumes has been known to reach the soil fertility. This is due to the fact that the concentration of nitrogen compounds in the soil increases as a result of presence of symbiotic bacteria. For this reason, normally nitrogen fertilizers are not needed in the fields cultivated legumes. Now let's know about the mechanism of nitrogen fixation. Inside the root nodules of leguminous plants, the bacteria proliferate. These bacteria exist in the form that has no cell wall. The bacteria of the nodules are capable of fixing nitrogen by means of the specific enzyme, namely nitrogenase. Now let's know about the nitrogenase. Nitrogenase is a complex enzyme containing two oxygen-sensitive components. Component 1 has two alpha protein subunits and two beta protein subunits and 24 molecules of iron, 2 molecules of molybdenum and an iron molybdenum cofactor. Component 2 possesses two alpha protein subunits and a large number of iron molecules. Component 1 of nitrogenase catalyzes the actual conversion of N2 to ammonia while component 2 donates electrons to component 1. Now let's know about legi- uh, leghemoglobin. A protein comparable of hemoglobin in animals has been identified in the modules of leguminous plants. Leg, uh, le- Hemoglobin, that is LHB, contains iron and is red in color. It is an oxygen binding protein. The heme part of leg hemoglobin is synthesized by the bacterium while the protein portion is produced by the host plant. Leg hemoglobin is absolutely necessary for nitrogen fixation. The nodules that lack LHB are not capable of fixing nitrogen. It is LHB that facilitates the appropriate transfer of oxygen to the bacteria for respiration to produce ATP, and energy in the form of ATP is absolutely required for nitrogen fixation. Another important function of LHB is that it prevents the damaging effects of direct exposure of oxygen on nitrogenase. As the oxy LHB supplies oxygen, bacterial respiration occurs. The ATP generated is used for fixing nitrogen to produce ammonia. The complex reaction is N2 plus 8H plus 8 electron minus plus 16 ATP gives rise to 2NH3 plus H2 as gas plus 16 ADP plus 16 Pi. Now let's know about hydrogenase. During the course of nitrogen fixation by nitrogenase, An undesirable reaction also occurs. This is reduction of H plus to H2, that is hydrogen gas. For the For the production of hydrogen, ATP is utilized, rather wasted. Consequently, the efficiency of nitrogen fixation is drastically lowered. It is possible theoretically to reduce the energy wastage by recycling H2 to form H+. In fact, some strains of Brasyrhizobium japonicum in soybean plants were found to use hydrogen as the energy source. These strains were found to possess an enzyme, namely hydrogenase. Recycle of the hydrogen gas that is formed as a byproduct in nitrogen fixation is now well known. It is advantageous for nitrogen fixation is the symbiotic bacteria possesses the enzyme hydrogenase. However, the naturally occurring strains of Rhizobium and Bradyrhizobium do not normally possess the gene encoding hydrogenase. Now let's know about genetic manipulations for nitrogen fixation. The nitrogen fixing bacteria that are closely associated with the world's food supply and are among the flavored organisms for genetic manipulations. Biotechnologists consider the following possibilities for effective utilization of diazotrophs as natural biological fertilizers. Gene alterations in Rhizobium species to improve nitrogen fixing efficiency and bacteria host plant interactions. Gene, gene alterations in Rhizobium species to improve nitrogen fixing efficiency and bacteria host plant interactions. Genetic engineering of Rhizobium species so that it can form a symbiotic relationship with non-leguminous plants such as wheat, rice, and corn. Transfer of genes for nitrogen fixation from Rhizobium species to other bacteria such as Agrobacterium tumefaciens. So, the so developed Agrobacterium tumefaciens can infect several important crops like tomato, tobacco, and petunia and fix nitrogen. Some other considerations include the insertion of nitrogen fixing genes into plants or even animals, including humans. This approach, which enables the plants, animals, and humans to directly trap and utilize nitrogen from the atmosphere, is rather theoretical. Life would be certainly different if introduction of nitrogen fixing genes in man can dispense with the habit of eating daily. Despite several possibilities and optimistic predictions, the success in the genetic engineering in relation to nitrogen fixation is very limited and briefly described. Genetic engineering of nitrogenous gene. Let's know about genetic engineering of nitrogenous gene. It is absolutely necessary to identify, isolate, and characterize the nitrogen fixing genes. NIF genes to undertake any kind of genetic manipulations. Genetic complementation is the common technique used for the isolation NIF genes. The procedure basically involves the identification and characterization of clones from a wild type library to restore nitrogen fixation in the various mutants of the original organism. The NIF genes were first isolated from the clone banks of the diazotroph Klebsiella pneumonia. This organism is found in soil, water, and human intestine, and its molecular biology is well known. The isolation of NIF genes by genetic complementation from K. petunia and involves, let's know. The first point is the wild type DNA of K. petunia, that is NIF plus, was caught with restriction endonucleases. A clone bank was constructed with a vector and maintained in E. coli. K. pneumonia cells are exposed to a mutagenic agent. This may result in the mutation of NIF gene to NIF cells. The NIF minus K petunia, K pneumonia cells are then conjugated with E. coli cells carrying the clone bank on a vector. The transformants of K pneumonia possessing NIF plus phenotype can be selected by growing the cells on a minimal medium that does not contain fixed nitrogen. The DNA fragment in the plasmid that contains NIF genes, which complements NIF minus mutation in K pneumonia, can be isolated. The isolation of NIF genes will be more effective if a series of independently derived NIF minus mutants are employed in genetic complementation experiments. This approach increases the likelihood of different NIF genes isolation. Further, by using DNA hybridization probes of NIF genes, DNA clone bank of K pneumonia can be screened. This allows the identification of additional genes that are involved in nitrogen fixation. Now let's know about nitrogenous gene cluster. The entire set of nitrogenous genes from K. pneumonia has been identified. Now let's know about some points. The nitrogenous NIF nitrogenase genes are located as a single cluster occupying approximately 24 kb of the bacterial genome. There are several distinct operands that encode 20 different proteins. All the NIF genes transcribe and translate in a well coordinated fashion. The NIF genes are under the regulatory control of two genes, namely NIFA and NIFL. Although the NIF genes have been characterized from K. pneumonia for technical reasons, the contribution of these organisms for biological nitrogen fixation is almost insignificant. However, the NIF genes of K. pneumonia have been used as hybridization probes to identify NIF genes from the DNA clone banks of diazotrophic organisms. It is now known that almost all the nitrogen fixing bacteria possess similar type of NIF gene clusters. Now let's know about manipulation of NIF genes. Some workers have been successful in introducing extra copies of nitrogenous regulatory genes, namely NIFA and NIFL, into diazotrophs. For instance, insertion of more NIFA genes into Rhizobium melioti resulted in an increased biomass production in alfalfa plants. This is due to the fact that nitrogen fixation in these plants is much increased. But the major limitation of this approach is that the genetically engineered bacteria have a diminished growth rate. As a result, these newly developed bacteria lose their efficiency as plant growth promoting agents. Despite continuous efforts by several groups of workers, no significant success has been reported in the transfer of NIF genes into plants. The major limitations now we will discuss. The transfer of 24 kilobase NIF gene cluster has been effective since the normal cellular concentration of oxygen would interactive nitrogenous enzyme. Any reaction in oxygen results in cell death. Transfer of one or two genes of NIF gene cluster is useless. There are no promoters that can respond to NIFA regulatory protein. Consequently, it is not possible to turn on the NIF genes in transgenic plants. The plant cells cannot process NIF gene transcript, which are multigenic in nature. The various regions now we discussed. Now let's know about the genetic engineering of hydrogenase gene. The role of enzyme hydrogenase in promoting nitrogen fixation has already been described. Nitrogen is synthesized by hop, that means hydrogen uptake genes, which are not present in the naturally occurring rhizobial strains. Considerable variations have been identified in hydrogenases from different organisms. There are different types of hydrogenases which usually contain subunits. Different genes code these subunits. Now let's know about the isolation of hydrogenous genes. The technique of genetic complementation can be successfully employed for this isolation of hydrogenous gene. HUP genes. In fact, HUP genes were first isolated from E. coli by this approach. Later hop genes from B. japonicum have also been isolated. Now let's know about the manipulation of hop genes. The hydrogenous genes have been introduced into R. leguminosarum. These Hoppler strains of bacteria, when inoculated into legumes, resulted in higher nitrogen fixation. Now let's know about genetic engineering of nodulation genes. Establishment of nodules on the roots of leguminous plants is a prerequisite for nitrogen fixation. Certain genes involved in nodulation, namely node genes, have been identified in Rhizobium melitoti. The technique of genetic complementation has been used to isolate node genes from R. melitoti. A large number of node genes, that is about 10, 20 to 20 node A to node X, have been identified as diisotropes. The node genes are broadly divided into three groups. One is common genes, second is host specific gene, third is regulatory gene. The functions of each one of the node genes in nodulation have not been clearly identified. Further, many more new node genes are being discovered every year. Now let's know about manipulation of node genes. The process of nodulation is complex through the participation of a large number of node genes. Besides various other factors, concentrations of nutrients, soil temperature, light, carbon dioxide concentration, etc. Despite attempts by several workers, no success has been reported to enhance the ability of rhizobium species for nodulation through genetic manipulations. Now let's know about the biocontrol of phytopathogens. Phytopathogens can drastically reduce the crop yield, which may be in the range of 25 to 100%. Chemical agents are commonly used to control them. This is associated with ill health effects on humans besides environmental pollution. There are certain bacteria that can lessen or prevent the deleterious effects of plant pathogens, fungi or bacteria, and thus they promote plant growth by indirect means. Plant growth promoting bacteria are capable of producing a wide range of substances that can restrict the damage caused by phytopathogens to plants. The important plant growth promoting substances are siderophores, antibiotics, and certain enzymes. Now let's know about siderophores. There are some bacteria in the soil that can synthesize a low molecular weight iron binding peptides, collectively referred to as siderophores. Siderophores have high affinity to bind to iron in the soil and transport it to the microbial cell. This is required since iron is essential and cannot directly enter the bacteria due to a very low solubility. The growth promoting bacteria through siderophores can take off large quantities of iron from the soil and make it unavailable for the growth and existence of fungal pathogens. This is possible since the siderophores produced by fungi have a very low affinity when compared to siderophores of bacteria. There is no harm to the plants since they can grow at a much lower concentration of iron in the soil. Now let's know about the genetic manipulations for siderophores. It is now clearly established that siderophores can prevent the proliferation of phytopathogens It is therefore logical to think of siderophore genes for more effective biocontrol of plant pathogens. Now let's know about pseudobactin. Pseudobactin is a siderophore synthesized by plant growth promoting bacteria Pseudomonas putida by genetic complementation and other techniques of molecular biology. At least five separate gene clusters involved in pseudobactin production have been identified. Now, genetic manipulation for improved synthesis of siderophore is not an easy job since it is a complex process involving a large number of genes. Some success, however, has been reported in cloning the genes for iron siderophore receptors from certain plant growth promoting bacteria and introducing them into other bacteria. Now, let's know about antibiotics. Certain antibiotics produced by plant growth promoting bacteria can prevent the growth and proliferation of plant pathogens. Some of the antibiotics synthesized by Pseudomonas are agrocin 84, agrocin 434, herbicolin, herbicolin, phenazine, omycin, pyrrolynitrin. Genetic manipulations for antibiotics. Let's know about genetic manipulations for antibiotics. It is possible to enhance the growth promoting activity of bacteria by inserting genes encoding the synthesis of antibiotics. In fact, the genetically engineered biocontrol bacterium of Agrobacterium radiobacter has been developed and is being marketed commercially since 1989. This transgenic organisms produces the antibiotic agrocin 84 and is toxic to crown gall diseases caused by Agrobacterium tumefaciens. By this biocontrol approach, the crop yield of almond trees and peach trees can be improved by preventing crown gall disease. Now let's know about the enzyme. Certain plant growth promoting bacteria are capable of synthesizing some enzymes that are degrade fungal cell walls and lyse them. These enzymes include chitinase, beta 1,3 glucanase, protease and lipase. Now let's know about genetic manipulation for enzymes. The genes responsible for encoding the enzyme namely chitinase and beta glucanase have been isolated and characterized. Chitinase gene chitinase gene has been isolated from Serralia morsensis are introduced into Rhizobium meliloti and Trichoderma herzianum. The genetically engineered organisms displayed as increased antifungal activity. By this biocontrol approach, the phytopathogen Rhizoctonia solani has been effectively controlled. Now let's know about biofertilizers. It is estimated that more than 100 million tons of fixed nitrogen are needed for global food production. The use of chemical or synthetic fertilizers is a common practice to increase crop yields. Besides the cofactor, the use of fertilizers is associated with environmental pollution. Scientists are on a constant look for alternate cheap and environmental friendly sources of nitrogen and other nutrients for plants. The term biofertilizers is used to refer to the nutrient inputs of biological origin to support plant growth. This can be achieved by the addition of microbial inoculants as a source of biofertilizers. Biofertilizers broadly includes some categories. Let's know about the categories. First is symbiotic nitrogen fixers. Second is asymbiotic nitrogen fixers. Third is phosphate solubilizing bacteria. Fourth is organic fertilizers. Now let's know about the symbiotic nitrogen fixers. The diazotrophic microorganisms are the symbiotic nitrogen fixers that serve as biofertilizers. Example, Rhizobium species and Bradyrhizobium species. The details on this bacteria with special reference to nitrogen fixation must be referred now. Many attempts are being made to genetically modify the symbiotic bacteria for improving nitrogen fixation. Now let's know about green manuring. It is a farming practice wherein the leguminous plants which are benefited by the symbiotic nitrogen fixing bacteria are plugged into the soil and a non-leguminous crop is grown to take benefits from the already fixed nitrogen. Green manuring has been in practice in India for several centuries. It is a natural way of enriching the soil with nitrogen and minimizing the use of chemical fertilizers. Rhizobium species can fix about 50 to 150 kg nitrogen or hectare per annum. Now let's know about the asymbiotic nitrogen fixers. The asymbiotic nitrogen fixing bacteria can directly convert the gaseous nitrogen to nitrogen rich compounds. When these asymbiotic nitrogen fixers die, they enrich the soil with nitrogenous compounds and thus serve as biofertilizers. Example Azobacter species, Azospirillum species. Now let's know about blue green algae, that is cyanobacteria. Blue-green algae multiply in waterlogging conditions. They can fix nitrogen in the form of organic compounds, that may be proteins, amino acids. The term algalization is used to the process of cultivation of blue-green algae in the field as a source of biofertilizer. Blue-green algae, besides fixing nitrogen, accumulate biomass, which improves the physical properties of the soil. This is useful to reclamation of alkaline soils besides providing partial tolerance to pesticides. Cyanobacteria are particularly useful for paddy fields. The most common blue-green algae are Azobacter species and Azospirillum species. Now let's know about Azolla. Azolla is an aquatic fern that contains an endophytic bacterium Anabaina azolla in the leaf cavities, providing a symbiotic relationship. Azolla with Anabaina is useful as biofertilizer, but due to certain limitations, the use of azolla has not become popular. First point is azolla plant requires adequate supply of water. It can be easily damaged by pest diseases. Azolla cultivation is labor intensive. These are the 3 points, three limitations of azolla. Now let's know about phosphate solubilizing bacteria. Certain bacteria, that is thiobacillus or bacillus, are capable of converting non-available inorganic phosphorus present in the soil to utilize form of phosphate, organic or inorganic. This bacteria can also produce siderophores, which chelates with iron and makes it unavailable to pathogenic bacteria. Thus, besides making phosphate available, the plants are protected from disease-causing microorganisms. Now, let's know about mycorrhizas. Mycorrhizas are the fungus roots with distinct morphological structure. They are developed as a result of mutual symbiosis between certain root inhibiting fungi and plant roots. Mycorrhizas are found in plants which are limited with nutrient supply. These plants may be herbs, shrubs, and trees. For the development of mycorrhizas, the fungus may be located on the root surface. In recent years, an artificially produced inoculum of mycorrhizal fungi is used in crop field. This practice improves plant growth and yield besides providing resistance against biotic and abiotic stresses. Biotic means pathogens. Resistant against biotic means pathogens. Abiotic climatic changes. Mycorrhizas also produce plant growth promoting substances. Now let's know about organic fertilizers. There are several organic wastes which are useful as biofertilizers. These include animal dung, urine, urban garbage, sewage, crop residues, and oil cakes. A majority of these wastes remain unutilized as organic fertilizers. These exist a good potential for the development of organic manures from these wastes. Benefits of biofertilizers: low cost and easy to produce. Let's know about the benefits of biofertilizers. First point is low cost and easy to produce. Small farmers are immensely benefited. Fertility of Second point is fertility of the soil is increased year after year. Third point is free from environmental pollution. Fourth is besides nutrient supply, some other compounds which promote plant growth are also produced. Example, plant growth hormones, antibiotics. Fifth point is biofertilizers increase physico-chemical properties of the soil, soil texture and water holding capacity. Sixth point is reclamation of saline or alkaline soil is possible by using biofertilizers. Seventh point is biofertilizers improve the tolerance of plants against toxic heavy metals. Eighth point is plants can better withstand biotic and abiotic stresses and improve in product yield. Now let's know about the limitations of biofertilizers. Biofertilizers cannot meet the total need of the plants for nutrient supply. They cannot produce spectacular results as is the case with synthetic fertilizers. Considering the advantages and disadvantages of biofertilizers, the realistic and pragmatic approach is to use combination of biofertilizers and synthetic fertilizers for optimum crop yield. Till now I have completed crop yield, that is a growth promoting bacteria in plants, how it works. I covered. Any questions regarding this you can ask me in the comment box, and subscribe my channel to know more topics of biotechnology and aware about biotechnological topics. Thank you. Please subscribe my channel. Thank you.